DNA replication starts with helicase. It will cut through the DNA molecule, splitting into two different strands. Topoisomerase then comes in and relieves the strain by breaking and rejoining DNA strands. Next, single-strand binding proteins attach themselves to the strands in order to prevent them from connecting. To make the new DNA, the already existing strands have to be matched to their complementary nucleotides. This is the job of DNA polymerase III, but before it can continue, it needs an RNA primer to place a template of nucleotides. DNA polymerase will follow the primer's example and complete the rest. It can only place nucleotides in the 5' to 3' direction. Following the DNA polymerase III is a sliding clamp that is binding the newly synthesized double helix. The second strand, or the lagging strand, is much harder to do, but it happens simultaneously with the leading strand. To replicate the lagging strand, the process is broken up into two segments, called Okazaki fragments. At the start of each fragment, there is an RNA primer. After the first fragment is primed, DNA polymerase III starts placing the nucleotides. Once it gets to the second primer, it detaches itself, and another DNA polymerase III attaches and finishes the sequence. Afterwards, DNA polymerase I revisits the strand and replaces the nucleotides placed by the RNA primer. However, since DNA polymerase doesn't bind the nucleotides together, it is the DNA ligand's job to do it.
Transcription. Transcription is the first stage of gene expression. Within the promoter is the start point where the RNA synthesis actually occurs. The eukaryotic promoters usually includes a TAT box, which is about 25 nucleotides upstream from the start point. In eukaryotes, there are transcription factors that are used to interpose with the binding of RNA polymerase and the initiation of transcription. Once the transcription factors are attached to the promoter, RNA polymerase II will attach to it. Both transcription factors and RNA polymerase II make up the transcription initiation complex. Once the transcription factors are attached to the promoter DNA and the polymerase is attached in the correct orientation, then the enzyme will unwind the two DNA strands and begin to transcribe the template at the starting point. Elongation. The RNA polymerase unwinds the helix of DNA, which exposes 10 to 20 DNA nucleotides so that the pairing with the RNA nucleotides can occur. This enzyme joins the nucleotides to the 3' and which adds to the RNA transcript. As the enzyme is moving along, the RNA is moving behind it. The new RNA begins to peel away from the template DNA strand and begins to reform a double helix with the non-template strand. The RNA polymerase II in eukaryotes will transcribe a polyadenylation signal sequence, which signals a specific polyadenylation signal AAU-AAA in the pre-mRNA. Once it is about 10 to 35 nucleotides downstream from the AAU-AA, the pre-mRNA is cut by proteins from the enzyme and the RNA polymerase II detaches.
RNA editing. A pre-mRNA needs to be modified in order to produce an mRNA molecule ready for translation. It starts with the 5' end receiving a 5' cap. An enzyme then adds 50 to 250 more adenine nucleotides, forming poly A tail on the 3' end of the pre-mRNA molecule. Pre-mRNA contains the coding regions exons and non-coding regions introns. The introns had to be removed before mRNA can be translated. RNA splicing cuts the introns from the region and connects the exons together. This produces a mature mRNA. All non-coding introns are spliced out by the spliceosome. Once the mRNA is mature, it will leave the nucleus and start translation. Translation initiation. The combination of mRNA-initiated tRNA in the small ribosomal subunit is followed by attachment of a large ribosomal subunit, completing the translation initiation complex. Elongation. An anticodon pairs with the mRNA codon in the A site. An RNA molecule of the ribosomal subunit catalyzes the bonding of a peptide bond between the amino acid of the new amino acid in the A site and the carboxyl end of the polypeptide in the P site. This removes the polypeptide from the P site to the A site. The ribosome translocates the tRNA from the A site to the P site. The one in the P site is moved to the E site where it is released. The mRNA moves along bringing the next codon to translate A site. This continues until stop codon reaches the A site. A release factor binds with the stop codon and hydrolyzes bonds with tRNA and the last amino acid in the P site.
This breaks the translation initiation complex.
Control of gene expression. Gene expression is the process by which a gene's information is converted into the structures and functions of a cell by a process of producing a biologically functional molecule of either protein or RNA. DNA is divided up into functional units called genes. Each gene provides instructions for a functional product. In this case, the functional product of a gene is a protein. A gene that encodes a polypeptide is expressed in 2 steps. In this process, information flows from DNA to RNA and finally to protein. Transcription. One strand of the gene's DNA is copied into RNA. RNA polymerase is the enzyme that controls transcription. A repressor binds to the operator and blocks attachment of RNA polymerase to the promoter, preventing transcription of the genes. A repressor protein is specific for the operator of a particular operon. When a repressor is bound to the operator, it blocks attachments of RNA polymerase to the promoter, preventing transcription of the genes. Thus, transcription is the common control point of gene expression in all organisms. Translation. During translation, the nucleotides of the mRNA are read in groups of 3 called codons. Each codon specifies a particular amino acid or PA stop signal. This set of relationships is known as a genetic code. Cancer is the primary effect of improper gene expression. It occurs when mutations in the genes that normally regulate cell growth and division in somatic cells occur.